Hello everyone, this is MD Sabid Ahmed. Today I'm going to talk about the preliminary kinetic characterization of F420 dependent glucose 6-phosphate dehydrogenase from Nocardia bacterium. At first, I'm going to start by giving some of the background information of F420 dependent glucose 6-phosphate dehydrogenase, especially from Mycobacterium. This enzyme has been studied extensively because of its health relevance to the tuberculosis disease. Photon-dependent enzymes are mechanistically different. FSDs, that like photon-dependent sugar 6-phosphate dehydrogenase, they are promiscuous towards other substrate. Like they can turn over multiple type of sugar 6-phosphates. So the major goal of our research is to characterize these FSDs using a steady state and pre-steady state kinetic methods to provide detailed mechanistic insight for these FSDs. This is the crystal structure of FGD from Mycobacterium. This crystal structure was solved by one of our collaborator, Dr. Gader Bashiri. It's a 78 kilodalton homodimer. Each subunit has one F420 molecule in it. And if you can see the extended view of the active site, there is tryptophan 44, histidine 40, and glutamate 109. All of these are in close proximity with the cofactor F420 and the substrate G6P. This model was done by docking studies. This is the structure of F420 cofactor. It's found in our enzyme. And for the FGD catalyzed reaction, the enzyme FGD is F420 dependent. The reaction it catalyzes the conversion of glucose 6-phosphate 6-phosphogluconolactone. So the FGD transfers a hydride to the oxidized cofactor, which then becomes reduced and it ends up with the oxidized product. So for the updated mechanism, so F420 bind first with the FGD forms a binary complex. This was based upon the inhibition studies, and then G60 binds with this binary complex and forms a ternary complex. Histidine 40 was acting as a active site base. Active site base, it was suggested. Did not act as active site base, it was suggested, but actually they act as an active site base by donating a proton to the glutamate 13. So they would facilitate the hydride transfer to the C5 position of F420 cofactor. Then it becomes reduced, and glutamate 109 acts as an active site acid by donating donating an proton to the N5 position, N1 position of the F420 cofactor. Then the reduced product is produced, and the oxidized product is released. For the pupil binding kinetic experiment, as my nocardia bacteria has F420 dependent enzyme, which is promiscuous towards other 6-phosphates, G6P, M6P, and F6P. So initially, I'm going to start using this condition. This condition has been used for Mycobacterium enzyme. Initially, I'm going to use this condition and I'll try to optimize this condition for my enzyme. Same thing will happen for pre-steady state and the steady state kinetic experiment. When I joined this lab, the gene was synthesized and inserted into the pVat vector. So when we got our result, we transformed it to the E. coli so that we can start our expression trial. For the expression and purification, so for the expression, we took the transformed cell and placed it in the agar plate, then add ampicillin and grow at 24 degrees centigrade for overnight. When the optical density reaches 0.6, we induce it with 0.01% L-arabinose and expressed for 36 more hours. So for the purification data, my protein is his-stacked, so I use nickel column to run this one. So I sonicate first, then centrifuge, then upload the upload the supernatant to the nickel column and ran the nickel column. After that, I check all the absorbance and make the absorbance photogram. And after that, I make the activity profile using the highest absorbance. And then I ran a 12% SDS space gel. You are seeing that I got a band at 56 kilodalton, which is my molecular weight. So I almost purified the protein, but it's still in working progress. I'm trying to figure out which condition is good for me. So I'm trying to optimize those conditions. At last, I want to thank my peer, Dr. Johnson Winter, and my lab mate, Ala Aziz and Jamari Howard.